Good morning once again. My name is Dr. Keke from the University of Lagos. I will be your compare today for this webinar. I would want to welcome us and also especially welcome those that really joined us quite early. Like we said earlier, we decided to give 5 minutes so that some persons who reached out to us are able to join. And I believe a lot of them have joined us, as I could see even in chat session. Thank you once again. So without much ado, we will be moving into the event for this morning. Even as we look at a scientific webinar on Africanizing Crispr technology for food and better healthcare delivery. We have seasoned researchers who will be taking us this morning on all that we need to grab. So please, I want us to pay up attention, sit back, and have a good learning. So to start with this morning, we're going to welcome one of our fathers and erudite scholar in the Department of Cell Biology and Genetics. The matter of fact, at the head of the Department of Cell Biology and Genetics in the person of Professor L.A. Ogukomi, who will be taking us in the opening address this morning. Professor, the mic is yours.
Dr. Ogojuku, let me welcome everybody to this organized by the Gene Editing Research Group of the Department of Genetics. I hope you are hearing me.
Yeah, we can hear you loud and clear, sir.
Oh, great. Thank you. So let me welcome all our participants online, and most especially our speaker for today. That is Dr. I'm trying to look at the name. Yes, Dr. F.N. Jomani. That name very well. The Director of Innovation, Entrepreneurship, and University, Lupe State University, Zimbabwe. Thank you so much for honoring our invitation. Our special regards to after Robert Mugabe, which is so popular in Africa, Mr. Dr. Emasi, I can't pronounce Nagawa. So much for accepting to give your own expertise idea on this program. Really welcome. The organizer, that is Professor Minari, who is the organizer and the head of the research for the Cell Biology and Genetics Department. So on behalf of the Vice Chief Host and the co-host, the Dean Faculty of Science, so I want on their behalf, welcome everybody to this webinar. And I believe by the time we finish, everybody will get 1 or 2 things in solving the problem of Africa. This is a very unique topic and it's to time. Especially in most Africa, we have problem of food insecurity caused by a lot of things. So I think very well and see how we can use indigenous to solve our peculiar problem. Nobody's coming here to solve the problem for us. So problem for ourselves. So we see how we can improve technology, try to use technology in our breeding program to give food security, probably precision breeding, and a lot of about us. You can also use this gene editing in the conservation of diversity. And from there, we'll be able to see climate smart agriculture so that all the problem of post-harvest losses, this and that, will be a thing of past. To see how we can add value to the kind of value chain to our food. I think the experts are the real experts to give us lecture today. So on behalf of the Dean and Vice Chancellor of the University of Lagos, I want to welcome everybody. And then most especially our keynote speaker, our guest speaker, F.N. Jomani. Thank you so much. I want to wish you a successful deliberation. Thank you and God bless you.
Amen. Thank you very much, our father, Professor L.A. Ogukomi. we really, really appreciate the welcome address. I believe this has set the pace for us this morning. As a matter of distinguished scholars, ladies and gentlemen, in this webinar, you've just heard the voice of Professor Elio Bukomi, the head of Department Cell Biology and Genetics, University of Lagos. As a matter of fact, last month I was under his tutelage when he delivered his inaugural lecture. It's a testament to the depth of effort he has added even in molecular genetics and also food security. Thank you very much, sir, for that welcome address. So quickly, we will be moving into the next session. Okay, so we'll be welcoming the Dean of Faculty of Science to take the opening remarks. However, she is ably represented by Professor Olusegun Ajayimbo Aye Jr. Sorry for that, please. She is ably represented by Professor Olusegun Aye Jr., the acting dean, who will be taking us this session of the opening remarks. Prof, the mic is yours. You are most welcome.
Thank you so much. Can you hear?
Prof,
can you hear you?
Yeah, we can hear a bit.
Loud and clear.
Yes.
Okay. Thank you so much. Our HOD, my very good friend, Professor Adebumi, the guest speaker, Dr. Jemani, I guess, the organizers, ably led by J.B. Minari, who has been so good in science and continue to be in. I appreciate you all and I welcome you to this dinner. Anything that has to do with our health, need to joke with it. Anything that has to do with food, joke with it. And one important thing about life is that there is an adage I say you eat. In this part of the world, food and all the rest are quite challenging. One of our little exposure, we are not as advanced as the other. And in most cases, we are not docile. We have the clock to ensure that in our little corner, whatever we are doing to help, and it is important for us to not gather together as we are doing today, and start a particular course in an attempt to in the technology that can enhance our food security and healthcare delivery. Important for us to note that nobody will do it for us as an African, and it is always very important for us and crucial at this stage that drought here and there, climate change, terrorism here and there. In most cases, we are running short. Whereas in other climes, they have excess. It is now our to look inward and internalize whatever we're supposed to do to enhance the good health of our people. And I know full well that you are well prepared for this webinar. And whatever we're able to pick today, we should now and let it rub on the society where we live. And on this note, to this webinar on behalf of the Vice Chancellor, Professor Ogushola, FASON, and my Dean, Professor Ashe. We welcome you to this webinar, and I wish you full interaction and a rewarding interaction. Thank you so much, Have a blessed day together.
Thank you very much, Prof. Yet, another inciting session from Professor Olusegun Ayejuwon. Thank you very much, sir. I believe our distinguished scholars in the house are geared towards having a nice time with us as you've nudged us this morning. Thank you once again. Okay, so quickly, we will be moving into the next session, okay, which is one of the presenters that will be taking us in this webinar this morning, okay, and permit me to read the citations before we move into the presentation. Dr. Fortune is the Director of Innovation, Entrepreneurship, and Industrialization at Lipen State University, Zimbabwe. He is also a senior lecturer in the Animal Science Department at Lipen State University and a former chairperson of the department. He holds a Doctor of Philosophy in Agriculture, to be specific, Animals Breeding and Genetics, from the University of Miyazaki, Japan, an MSc in Agriculture, Animal Science from the same university, and a Bachelor of Science degree in Agriculture from University of Zimbabwe. Fortune's experience and expertise is in animal science, bias towards animal breeding and genetics. He has co-published 30 research articles, including several book chapters, peer-reviewed articles, and international conference proceedings. Dr. Fortune Jomani has published a lot of research articles, like I mentioned earlier, and some of his research interests and ongoing projects include livestock breeding and genetic improvement, the application of biotechnology in animal science, and the application of artificial intelligence and machine learning in animal production. Distinguished scholars, join me as we welcome our first presenter for today, in the person of Dr. Joman Fortune Entawa. We can see your screen, Dr. Fortune.
Thank you very much. If you stop sharing, I can start sharing my screen.
Yes, we can see your screen now. You're sharing already.
This one sharing.
I'm not sharing. It's being shared on the other side. If you stop sharing, I can start sharing. Thank you. My screen is coming up. Kindly confirm that you are now seeing my screen.
What is my name on the screen? Yes, I can.
Thank you very much, colleagues. I would like to appreciate the University of Lagos. Let me just turn on my camera so that it's a bit. I would like to thank the Vice Chancellor of the University of Lagos, the Dean of Science, the Head of Department, Cell Biology, my co-speaker, Professor Joseph. I want to appreciate those that have managed to join. I really appreciate. I think introductions have done, so I'm going to my presentation. The outline of my presentation, I'm going to talk about some key milestones in Crispr technology, which have been attained for the past several decades. So have an appreciation of when this technology began.
Sorry, Dr. Fortune, can you hear me?
Hello.
Yeah, please kindly go on full screen.
Okay, thank you.
Welcome.
After that,
perfect.
Status and crop improvement. Since this webinar is also touching on food security issues. Then after that, we'll talk about the possible role of CRISPR in food security. And what strategies as Africa can we adopt using this technology so that it can speak or alleviate challenges or opportunities that we do have as a continent. of what has transpired, CRISPR, which is clustered randomly spaced palindromic research, was really identified in 1987. Then in 2000, there was an whereby the recognition of CRISPR in prokaryotes was noticed. Then 2 years later, CRISPR was actually coined, and Cas genes were defined. Then 2005, the of CRISPR in terms of as an immunity mechanism was also noted. Then the first experimental evidence of CRISPR noted in 2007. Then in 2008, CRISPR, its effects on DNA was actually recorded. Then 2009, CRISPR complexes could cleave DNA. I think there's some background. You can mute some mics. I think there's some interruption. In 2010, Cas9 guided spacer sequences actually cleave double-stranded DNA. So those are some of the major events in this technology. The scientific improvement. My speech is focusing on what possibly can we use in terms of applying it in our African context. So the current status in most of our countries is that most crops and livestock are smallholder sector, and these are the small farmers, some subsistence purposes, some most of them have land or limited resources. So there is low productivity in that sector, and that's what we call it the smallholder sector, which is common in most of our African countries. Then there's the issue of limited genetic movement, particularly in that sector. And that sector is very important because that is of people on our continent. There are some countries ranging from maybe 40% to 60% being a rural population, and most of them are depending on farming or agriculture as a way of livelihood. Those are the challenges that are there. Then the pictures are just the differences between the smallholder sector and the commercial in Africa and also elsewhere. I think there's a middle picture which is showing two small chicks. I've put an arrow there in the middle of the slide, demonstrating to a farmer. A farmer was thinking that the different that we have in our indigenous chickens compared to commercial in the commercial sector is because of feeding or management. It was a practical demonstration to the farmer. I gave him some chicks. Some of them were not genetically improved. Those are the typical chicks that you have in villages, and the other chicks are chicks that most commercial farmers or companies and feed them. And usually these ones in 6 weeks or 8 weeks. So I was trying to prove a practical point to a farmer that it's of genetics that is really involved. So these are the chicks, 2 old. Then after about 3 weeks, they were being taken care of in the exact same environment, same feed, given ad libitum. That means there were no restrictions of feed, but they were exactly in the same environment. But after 3 weeks, those are the differences. One is the picture at the bottom there, now 3 times bigger than the other. It just shows the power of genetics and how it can affect productivity. The other picture that is showing 3 is just highlighting the changes that have happened in the commercial sector, whereby in the early 1950s, that is the of broilers that were there at 8 weeks old. That is what could be achieved. And kg, and if you look at that first chicken on those 3 chickens that are there, most people, if you give them an offer that I have this chicken, can you eat it? Be comfortable to have that because it doesn't look mature or large. Some years later, in the 1970s, they could clock 2 kgs, be comfortable to eat that chicken in the middle. And now the broilers can achieve more than 4 kg just in 8 weeks. And this highlighting the power of genetics. And this was achieved traditional breeding practices that we call selective, which involves taking the best candidates in a population, making them parents of the next generation. Doing that, so every year or every generation, you are actually changing the population genetics, selecting individuals with the best performance, and definitely those individuals with the performance are individuals that have the better genetic growth rate, better feed conversion efficiency. That means the maximum they eat. So that is the transition that has happened. But has been part of this transition, as I highlighted earlier with these people. Most of the poultry in our smallholder sector is deliberate, aggressive genetic improvement to attain high productivity. That's why we are buying from commercial companies, and poultry actually on the African continent, that is in the commercial sector, imported as either as eggs, flies in. Then those eggs are now used to produce grandparent and parent stock, which will now produce eggs or chicks that are sold to us for 6 or 8 weeks and sell. That is that sector. But as I said, most of our village and smallholder sector, the indigenous chickens are breed, and the productivity is low. And we are now talking about an Agenda 2060 achieving higher income status for the African continent. And these are issues that we need to deal with. It doesn't only concern crop production, so even concerns other sectors like crop production. In most of our staples, staple crops like maize, millets, some have yams and potatoes. So we find that in most traditional crops, there has been limited genetic this is just highlighting animals, but the situation is true for the crop sector in the smallholder. Then the issue, we are also cognizant that yes, there are also that are there, but these are very limited. You find that maybe only 5-10% on the country of the population are actually big commercial access to commercial breeds. The other picture is also genetic improvement, again, done traditionally or classical animal breeding, whereby people look at animals, look at the phenotypes or the characteristics, select those that look better, breed them again. Again, this is highlighting an animal called it does have a genetic defect, and what is commonly referred to double mastering is a condition whereby there's regulators of growth. It's a genetic defect, and it results in animals that may have 30 to 40% mass than an average animal. But that's an advantage or a trait of economic importance, and it has been achieved through selecting animals. Then to highlight again, so that we have a context of where we are coming from in terms of movement and where we are going. Most of these traits that are quantitative in nature, low heritability, relatively low heritability, particularly those related to reproduction. For example, on cattle, things like milk-cute, things like litter size is just 5% plus or minus. Publications will give you figures, but generally for reproductive traits, the low. When we say heritability will be low, basically we are saying maybe it ranges from 1 to, in terms of percentage, and maybe up to 20%, saying the whatever we're looking at in that particular be it crop, 20% of it is influenced by additive genetic factors transmitted from parents to offspring. That's why these parameters are useful. I'll relate to these figures as I proceed with my speech. Traits that have low heritability can be good candidates to be improved CRISPR technology. So because with selective of time, it's slow because the influence of genetics is limited. That's when you that's when you have to look at other strategies in terms of how can you achieve so that is the context of the status genetic improvement situation in Africa. So in a nutshell, we have of our population in the smallholder sector, in rural areas, traditional breeds of livestock with open pollinated varieties of traditional ones, which may not be as productive. Been very limited systematic genetic improvement processes. So with that background, I think we can CRISPR to try and improve this situation. So the CRISPR now, with this technology that is coming in and this and around it, we have done a lot of work within the continent. I've just taken one table of one of the public to illustrate a very important point. There's a lot of information that a lot of African researchers on the continent and outside the continent looked at what we call genetic markers to try and improve make importance. With this particular one, I worked on the growth hormone physics, which are just a change in a certain gene in its alternative versions of it in terms of sequence. We call polymorphism. So I looked at polymorphism in the growth hormone. This was during my PhD, Japanese black animals. Just to highlight that you did and how we can try to link or harness such board of information continent and outside the continent to push our agenda of crop improvement on the continent. I'll take you to the second last row. So these are the genetic polymorphisms, various groups of the, we looked at in brackets, those are livestock numbers, AA group at like 55 animals and so forth and so on. And CC only animals with that particular genotype. But in terms of results now, at carcass weight, these animals were steers slaughtered, their carcass weight was evaluated because the interest for us definitely is all of the meat which the farmer is going to sell, which the customer is going to buy. If you look at such a genotype as AA, in terms of carcass yield, it gave around 480 kilograms. Then a group AB 463, AC 461, a group like a BB genotype for 469, BC at 464. Then the CC genotype now, though the animals were limited of around 509, which is almost 30 to 40 kgs compared to other genotypes. So where does CRISPR coming is now we have a lot of these publications within the continent and outside. We know the growth hormone, we know the sequence of the growth, the polymorphisms that are in the growth hormone. Now, I think it can be a strategy to now infuse these genotypes, natural occurring phenotypes. Now we can to harness CRISPR so that we infuse harness CRISPR, use these maybe at egg level, fertilization, try to use CRISPR at that stage, then put genome editing, put such a modified egg back into the animal, and then that offspring that comes.
Sorry, Dr. Fortune.
Favorable genotypes. Hello?
Yes. Are you still sharing your slides?
See my slide?
Yeah, we can see your slide for some minutes.
Thank you. Thank you for letting me. Okay, this slide is coming up. I think there was a glitch. Kindly confirm when you're seeing them.
Okay, yeah, it's coming up. Not yet.
Kindly confirm that you're seeing my slides.
Not yet. Oh,
Okay.
it's coming up. Okay, yeah, perfect. Perfect. Thank you.
All right, thank you. So the what I was talking about right now is this particular table of some work that we did, a growth hormone, which has various polymorphisms. So I was just, the growth hormone has various polymorphisms that we looked at, and these affect carcass. And some of the things that we observed is that some genetic have a lower carcass yield, which is 30 to 40 kgs below scoring genetic groups. Why is this important? We think about harnessing or utilizing CRISPR in crop, animal, or food production. There is this background work that we have already done. Used information about sequences that are there, polymorphisms that are there, to try to use CRISPR now to induce or these genotypes in, particularly to improve traits of economic importance, such a genotype. And if there's a 30 kilogram or a 40 kilogram, if a farmer has 100 animals, that translates to 400 kgs. And if you consider that at national level, the countries slaughter millions of animals, and that would be tens of millions of kgs in improved productivity. And now,
Hello, good afternoon, everyone. I just want to confirm if our speaker is having network challenge.
Yes, we can hear him.
Okay, thank you very much. So
I think he's gone. He's gonna.
Okay, okay. Sincere apologies for this. So I'll reach out to him and let's see if he's able to get back on track. Thank you.
hello, can you hear me now?
Okay, now we can hear you.
Can hear you.
I'm not sharing your screen yet.
Thank you very much. Sorry, there was a power cut, so I had to change to.
Oh, okay.
Can you see my screen now?
Yeah, it's coming up.
Okay. Thank you very much.
Very well, perfect.
Let me wrap up.
If you can do full slide, please.
Thank you. You can see the full screen right now.
Yes. Great.
Thank you very much. So I just wanted to highlight that there's a lot of background research is available that we've done. And now we need to think about using those marker-assisted selection, the knowledge that we have about sequencing polymorphisms. Can't we take those polymorphisms, use to induce those polymorphisms in our populations? And that frog, as I highlighted earlier on, who had been left behind with classical animal and elsewhere. So that is what I wanted to bring to attention. Of muscle steatosis, which is a condition whereby muscle turns fat. It's also something that we looked at, and we have researched on that in terms of genetic polymorphism that influence that we can try to rectify in our animals. Then this picture is just the traditional village we do have, and improve the breeds of pigs there. So we foresee that with a genetic improvement, it can transform the life on our continent. Then in terms of without a strategy, really, we cannot go anywhere. We can have CRISPR technology. We don't have deliberate efforts to actually utilize this technology, can be left behind with this technology, while other countries are more. So the first critical element is infrastructure. Definitely the good laboratories for us to work through, among other things, even power supply for laboratories, power backup. Those are critical elements able to use these technologies. So it's quite things that need to be aligned in terms of infrastructure for us to fully utilize this. Then there's of human resource development and retention. Yes, we are training University of some people, we are training some people here, and all over the continent there is training and development happening. But unfortunately, most of our countries suffer from a break whereby the bright PhDs that we produced, bright MScs that we produce, they are now absorbed outside the continent, maybe greener pastures. For our talented staff members, deliberate efforts to retain. Then there's also the issue of research also needs funding for it to go on. There are commitments. I know most of African countries have subscribed or agreed at least 1% GDP funding for research and development. But in terms of actual questions, find that some countries or a lot of countries are falling short. If you countries, some do 2, 3, 4% of GDP, but already they are developed. So to leapfrog and be able to adopt these technologies, we need to have a commitment in terms of budgetary support and private sector. Then we tap into existing body of knowledge. As I said, there's happened within the continent and outside the continent that we can harness as we try and use CRISPR to improve food security. There's also highlight potential economic benefits for the private sector. Private sector support is relatively low in terms of the compared to other regions. Private sector funds from an agriculture person perspective, and I've just put also a slide also a good summary that we did called genome editing and gene drives available on online for people that might be various genome editing tools in a summary format. That ends my presentation. Thank you very much.
Thank you very much, Dr. Fortune, for that insightful section. Well delivered. Apologies for the network glitch. It's something that we do not have power over, but quite great that you were able to manage that. So thank you very much, and I believe our distinguished scholars, ladies and gentlemen, and researchers in the house have been able to, assimilate a whole lot of information. So I want to crave your indulgence, participant. Please, if you do have questions, please you can drop that in the chat box so that we'll begin to take the questions. So all questions would be addressed at the end of the second presentation. All the questions would be taken. Okay, so please kindly go on full screen. So quickly, we will be moving to the next presentation. And to take this presentation is also one of our erudite scholars in the department. So I will be welcoming my colleague, Esosa, to come and take the citation of the next speaker. Thank you.
Thank you very much, Dr. Ugo. So I've taken a citation of Professor J.B. Minari. J.B. Minari is a professor of Faculty of Science, University of Lagos. He's a scientist with specialized skill on cell and molecular biology, genome editing, and DNA forensic. He started his university academic career about 2 decades ago, in which he has mentored and supervised over 100 students at both undergraduate and postgraduate levels, which have worked in his research space. He has authored over 70 publications in reputable journals, both locally and internationally, and some are still in press. He has a membership of professional and academic groups where he has served at various offices. These include member, Nigerian Society of Biochemistry and Molecular Biology, member, Biochemical Society, United Kingdom. He has facilitated and attended several trainings and workshops, among which are 2 consecutive DNA molecular biology training and workshops organized in the Department of Cell Biology and Genetics, University of Lagos, in collaboration with Cummings School of Medicine, University of Calgary, Canada. Professor Minari has won and managed several grants, amongst which are UKGCFRONF98EWS COVID-19 grant awarded by the United Kingdom Research and Innovation in 2020, Tedford grant awarded by Tedford Federal Government of Nigeria 2020. He has also attended several conferences, both local and international, where he has presented his research work, served as plenary speaker and parallel section moderator. Kindly join me as I welcome Professor J.B. Minari.
Yeah, thank you very much. Let me share the screen myself. Before I continue, permit me to appreciate everyone for joining. Our head of department, the Dean, ably represented by Professor Olusegun Ayejuyo, for creating out time to be with us, and specifically my friend, Dr. Jamal Fortune, all the way from Zimbabwe, that actually played a host to us in 2022 when we came for the genome editing training sponsored by Gene Convene, and Trend Africa. Thank you very much for creating out time to share your wealth of knowledge with us. Dr. Fortune, thank you very much. We sincerely appreciate your time for that wonderful petition. Okay, so for a few minutes, I want to take you through. Okay. So let me put on my stuff. So I want to take you through an aspect. For this webinar, we are looking at two aspects. One is looking at the food security, which Dr. Fortune has actually dealt with. I will be taking you on the health aspect of it. So I will be speaking on, sorry, I hope you can see my face now.
Your video is on now.
Thank you. So I will be speaking on Africanizing Crispr technology for us to have a better, effective healthcare in Africa. Okay, so let me start to share with you that in Africa, the healthcare system, of course, is faced with a series of challenges, among which we have issue of underfunding. You agree with me? We have issue of lack of skilled workers. The few ones that we have also traveling out of the continent, poor leadership and budgetary support for the healthcare system, and also we also have issue of non-availability of latest technology for healthcare support. And that is probably what's going to guide us more in this presentation. And that's why you see that most times when there are a little bit of serious or complicated issues, people are always flown out of the continent. Now, let me just share with you some of the major health burden in Africa. In 2009, when the ECO, SOC, UNESC, WHO had a ministerial meeting in Doha, Qatar, they came up that under non-communicable diseases in Africa, that cardiovascular disease, diabetes, cancer, chronic respiratory disease account for about 60% of death globally. And you will see that this huge burden, the magnitude has a lot of socioeconomic impact here for us in Africa. So it's a major, huge burden in Africa because these categories of non-communicable diseases has actually led to death in Africa. So they even made a projection to now say that in 2030, there's going to be an increase in death in some of these non-communicable diseases. And one of them that was actually pinpointed is cancer. And so, you agree with me that steadily, we are just entering 2025 in a few weeks' time. You can see in 2025, it's still increase in cancer. We're going to look at that a little bit more. So that's been the projection of that meeting held in Doha, and that is actually affecting us in Africa. Now, what are the major setbacks we have in Africa healthcare? We have patients uninformed. That is very, very key. A lot of things happen when people have some of these ailments, they are not well informed. They don't even know where to go. They don't know the right health system to actually look for. Then lack of funds for new and also to maintain infrastructure. Then expensive costs of treatment is also a major problem in Africa. The inadequate number of properly trained and compensated staff, and because of that we have a capital flight, we have people moving in drones out of the continent, and then poor diagnosis as well is also a major problem, which we can actually as scientists rise up to the occasion to actually try to solve. Yes, we've enumerated some of these setbacks, we've enumerated some of the challenges that we face, but the point still remains that in spite of all these challenges that we face, Africa healthcare system, we have the potential to actually bring solution, innovative solution, actually still lies within us in Africa. And this could be done when we are able to Africanize the latest technology. Africanizing the latest technology means that we have our own technology that has been there in the past from our fathers, but we need to up our games. So we need to Africanize latest technology. I discovered that in Africa, when we have latest technology, it seems we have some resentment against it. Oh, this one is, they want to kill us. This one is not good. This one is not good. And it's actually pushing us back. Don't forget what I said. Part of the setback is, we have poor diagnosis, and then we don't embrace new technology as quickly as possible so that we can move with the rest of the world. And one of the latest technology that we should actually flow in now and try to domesticate in Africa, try to Africanize in Africa, is the Crispr system, is the Crispr technology. So when we talk about the Crispr technology, what do we mean generally when we refer to the CRISPR technology? Of course, we know that the word CRISPR means the cluster regularly interspaced short palindromic repeats that was discovered by scientists in bacteria, that those bacteria actually use them for defense mechanism. And now this CRISPR is now being used now to ensure that we can cut some section of the genome, we can correct those aspects of the genome, and that can help us in treating some unimaginable diseases that in the time past we felt they don't have any remedy or they don't have any treatment. So with the help of a guide RNA, the Cas9 protein can cut, and then you can replace the sequence that you want, or you can just allow it to merge, to join on its own. And so if we are able to if you are able to appreciate or embrace this technology, what's going to do for us, the advantage or the benefit is that, number one, is going to improve on the diagnosis. So we're going to have accurate diagnosis. Let me tell you that we still have so many diagnoses now that gives false positive or false negative. And then when people, and we're in Africa, and you go to the hospital and then there are some diagnoses and they tell you that, oh, we didn't see anything. And then you say, oh, it's the village people that is after me, not knowing that it's actually poor diagnosis that has actually happened. So embracing CRISPR technology can enhance accurate, give us accurate diagnosis, and not only accuracy, it can also make it fast. Okay, there are some diagnoses that, as I'm going to explain to us, that CRISPR can make some diagnosis to be fast. Then also, CRISPR can also make diagnosis to be easy. There are some diagnosis that is done now that is very cumbersome. You have to extract from here, you have to take there, you have to move there, before you get a set of results. But with CRISPR technology, it can make diagnosis to be easy. And then another major advantage of this is that you can achieve your targeted therapy, which this can help to treat so many disorders that has been known in the past that cannot be treated, like cancer, like diabetes, cardiovascular disease, and other things, other diseases disorders that are just being managed can actually be treated using the CRISPR technology. And this we need to domesticate here in Africa. Okay, let me just give you example. For example, now under diagnosis, CRISPR for diagnosis. For example, we can use CRISPR for COVID-19 diagnosis. You remember a few years ago, precisely late 2019 to 2020, there was emergence of the SARS-CoV-2 virus, which ended up to be a pandemic, and there was a whole lot loss of life all over the globe. And then there was panicking everywhere. People don't really know what it is. But eventually, they came up to know this is how it spreads. It can come from the bath, it can come from consumption of from food, from contamination, from human to human transmission, and so on and so forth. But having said that, one of the major challenge then was getting the diagnosis. So getting the diagnosis was a major issue. So there was a lot of diagnosis that came up, like the body measurement of the body temperature. If you remember, entering major places and they want to take your temperature, you know, there was also issue of chest CT. Do a CT scan of your chest to see whether they can get some spots for you to say, oh, can we know whether this person has been infected. There was also the lateral flow assay that was also used. Some people use biosensor to also check some of these things, to check the antibodies. The WHO even came up to announce some genes that can actually be used to analyze the SARS-CoV-2. And so the PCR technique was one of the main being that was generally acceptable all over the world, with some level of accuracy to be able to have a right diagnosis, with getting samples from different portions, from the mouth, from the nostril, from the blood, and so on and so forth. Now, that was what actually the WHO came up with. But you see, some of these techniques came up with some challenges. For example, a PCR sometimes could be insensitive. So we have inadequate sensitivity to detect asymptomatic and pre-symptomatic patients. So in this particular instance, for example, I remember that during this COVID, when my citation was read, we won a grant, UK ROI, for us to monitor the waterways for early signals. And I know part of what we were checking from those was those selected genes. So I remember in one of the work, when we were doing it then, we discovered that one of the genes was actually missing in a part. So we were like confused to say, I now told them that let's document it that way. It was thereafter, after some days, it was announced that there was a particular strain, okay, that probably that portion of the gene we're even looking for, you understand, probably because of the the frequent mutation of the virus, that particular portion has actually been deleted. So sometimes because of that, it could make your PCR technique to be a little bit insensitive in some cases. Also, the PCR method also requires, of course, laboratory settings, dedicated facilities, equipment, trained personnel. I remember that during the peak of it, you were looking for personnel as well. So these are some of the shortcomings. And also, it was also time consuming. If you remember that as the COVID was passing and the countries were requesting for COVID results before you can travel out of the country, people will submit their samples and they won't get it in days, in weeks, you know, especially in Africa here, before they travel. So these are some of the issues we have with diagnosis. But do you know that CRISPR can solve all these problems? You know, with CRISPR technology, you can enhance the sensitivity of your diagnostic tests. For example, the Cas12 and the Cas13 can actually be used for this COVID diagnosis. Okay, and what can you do with that one? When you get your test tube and then you get your samples in that contains the sample, maybe contains COVID-19 or not, the SARS-CoV-2 or not. So what you now have there is that you can have if there is the virus there, you can get the the CRISPR-Cas13, okay? The CRISPR-Cas13 can actually split on some portions of the viruses. When it splits on portion of the viruses with reporters. Of course, when you consult them, you consult them with reporters. So the reporters helps you to bring change of color that can make you know that, oh, something is happening here. So when you have caught some selected region, when you have designed your guide RNA to target some sequence on the virus, okay, with your reporter. So with that, it can now make, your reporter will now bring out that change in color. So immediately there is that change in color because the viruses are there. Definitely, you are going to have a change in color in your test tube, and that tells you that gives you the presence of the virus in the samples that you have collected. So with this, you can actually enhance your diagnosis of the CRISPR. And not only that, what another major advantage of the CRISPR-Cas13 is that you can target multiple sites. You can design several guide RNA targeting several regions, of the virus. And so when you target several regions of the virus, it enhances the sensitivity. And after that, you can now amplify. And with that, it can really give you a very a good result in terms of sensitivity. And also, the CRISPR technology you can also be made very fast. So in this particular case, you don't need to be extracting the RNA, you don't need to be doing the cDNA, you don't need to be quantifying. Just if, for example, what was envisioned with the CRISPR-based simple diagnostic test is that you can just prepare it, okay, into a device like this, so that when you get your sample, you put it within few minutes, you can get your result. Or you do a load into a one-port reaction tube. When you have prepared all your guide RNA, everything has been there. So just get the sample from the patient, just put it there, and within a few minutes, when there's a change of color, your result is already out. So with this, it can actually enhance our diagnosis, to make it easier, to make it faster, and then to make it accurate, because you can target several, so it's going to increase the sensitivity. Okay, another aspect that the CRISPR can enhance it can enhance the healthcare delivery is also by treatment of diseases that we once thought were not treatable. A typical example is the sickle cell disease. I think you are aware of the recent drug that is already being in use already in some parts of United States and United Kingdom, called Casgevy. And you see, and that is what I said, that Africa is a little bit slow in embracing the latest technology. Now this technology is being used outside the country. We are outside the continent, not even the country. So we are not yet there. It's still like far away from us. And that is why we should Africanize some of this technology. Okay, we should Africanize some of this technology. Let me explain briefly what they did with that. We remember the sickle cell disease that has to do with a change of a nucleotide on chromosome 11. Okay, so when you have your GAG that's supposed to code for your glutamic acid, and that should be responsible for the proper hemoglobin, that will lead to the normal cells that's supposed to be disc shape. Okay, so that should be the normal thing. But in this particular sense, we have a situation whereby then that there's a change of that nucleotide. So instead of you having your GAG, you're having your GUG. And instead of having your glutamic acid, you're having your valine there. And that is leading to you having a malformed hemoglobin, which is sickle shape, and of course cannot perform the function. So you now have a sickle cell. Okay. Yes, we have available way to manage it. We have medication. People use hydroxyurea that will just help you to boost your immunoglobulin production, but that does not solve the problem. It's just to manage it. When people do blood transfusion, at a point we're doing bone marrow transplant, which has also tried, but comes with sometimes a lot of side effect. And then in Africa, a lot is being done in terms of herbs, alternative medicine, medicinal plant. They are just there to ameliorate. They cannot solve it. If anybody tells you that plants will completely solve sickle cell disease, that person will be deceiving you, because it's a genetic disorder. So except you correct that particular genetic disorder, it will just be there. So we'll just be managing the situation. So it's either the person will just be placed on a permanent drug, you'll be watching when you have crisis, and so on and so forth. But if we're able to domesticate this, Africanize some of this latest technology, is actually going to help us. So, and what have they done on CRISPR now? On that Casgevy, it's not only Casgevy, we have other drugs that have also been discovered along that line. But let me just explain quickly what they did on Casgevy. So, normally there's what called the fetal hemoglobin. That is the hemoglobin responsible 6 weeks after pregnancy. That is what the baby feeds on through the fetal hemoglobin. And after delivery also, some 3 to 4 months, the fetal hemoglobin is the one that the baby actually uses after delivery. But something happened. There's a transcription factor called the BCL11A. What that does is that it helps to suppress the expression of the fetal hemoglobin. And when it does that, that is when the normal adult hemoglobin will begin to start expressing. So, but in the case whereby the adult hemoglobin is already mutated, like we explained earlier, so there's going to be a problem, because the hemoglobin is going to lead to a sickle cell. So, and what have they done with CRISPR? What have they done with the CRISPR technology? So they use the CRISPR technology to target the BCL11A transcription factor, okay? And so they just disrupted it, okay? Just cut a portion. This is the palm, and then this is the palm sequence, and then they target this region, removed this particular region, brought in another sequence that will not allow the BCL11A to express. So by the time you suppress the BCL11A, what's going to happen is that the HbF is going to find expression. So there will be a lot of expression of the fetal hemoglobin. And when you have expression of the fetal hemoglobin, the sickle cell patient will now, instead of having the HbA, will be having the HbF. And the HbF has a higher affinity for oxygen. So it's going to bring that person back sound, better, and it's going to completely correct it. You don't need to be taking herbs. You don't need to be taking hydroxyurea. If you look at the patients that they've used them on it, few days they are off the hospitals, and then their eyes became very clear, and they became sound, and so on and so forth. So these are the kind of technology that we should encourage in Africa. Yes, we need to move on. We don't need to just stay on it. And I'll say, why do you think we should domesticate CRISPR in Africa? Do you know that Nigeria is regarded as the global epicenter of sickle cell disease? Do you know that Nigeria has the highest number of sickle cell patients in the world? Do you know that about 35,000 children, under 5 die each year, taken from 2003 to 2013. So imagine from 2014 to 2024, even if it is the same margin that is dying each year, with all hydroxyurea we are using, with all herbs that we are using. So all these, they're actually affecting and leading to a whole loss of life. And this is not peculiar to Africa, Nigeria alone. It's all over Africa. Africa is badly affected. Nigerian number is higher because our population is high. Our population is about over 200 million. Okay, so if we were having 35,000, African countries that their population is 20 million, they might also be having 3,005. So they might think that their own is not low. It's virtually the same. So just because our own population is high, that is why our number is high, and we are being seen as the epicenter of sickle cell disease in the world. So that is why we need to CRISPRize healthcare in Africa. I coined that word CRISPRize. We need to take this CRISPR all up in Africa, and we need to domesticate it. We need to Africanize the CRISPR technology. Number three, another way we can actually, again, use the CRISPR to enhance the health delivery is on cancer treatment. Okay, of course, issue of cancer when we have malignancies of cell, uncontrolled growth of the cell, which has a lot of interplays of the genes. Look at these statistics as at 2021. To our population over 200 million. Cancer death each year is still estimated about 78,000 every year, and about 124,000 is being reported each year. And of course, breast cancer is leading the pack here with 37%. So we have other forms of cancer also, like prostate cancer in male, that is also on the increase in Africa. And most of the time, what do we the available treatment and management that we have surgery, we have chemotherapy, radiotherapy, immunotherapy, stem cell transplant, photodynamic therapy, lasers, blood product donation and transfusion. We also have alternative medicine where we use herbs, we use diet, we use supplements, we use vitamins, and so on and so forth. Now, these are some of the available means that we've been using to manage some of these things, which most of the time is just to manage. Still, even all over the world, issue of cancer is still almost like death sentence, except there is God's intervention. So, but do you know that CRISPR technology can actually address some of these issues? So CRISPR can combat this cancer by targeted therapy, okay? And those cancer, and it's only the cancer cells that you can actually target and see how you can get them eliminated. Now, in our lab in University of Lagos here, we are venturing into that, to working on using the CRISPR technology to actually address some of these genes. Like I told you, that when it comes to issue of cancer, it has to do with a lot of interplays of the gene, depending on where you want to focus on. We have different areas that you can use the CRISPR to actually edit. You can use it in disruption, you can use it in knockout, you can use it in targeting. So you can target a particular sequence, cut it off, and replace it with the correct sequence. Okay, you can just disrupt the sequence. Okay, that might give you a particular result that you want. You can decide to knock out a particular gene to achieve what you are going to do. So we are actually looking at some of these genes in our lab currently in the University of Lagos, and trying to use CRISPR to actually work on it to see how far we can go in making sure that we have a solution to it. So like I said, we need to CRISPRize healthcare in Africa. Yeah, CRISPR has a whole lot of applications in health that it could be very, very beneficial to us, like I said, in diagnosis, and also in treating some of these genetic disorders that we think it can never have a solution, but now solution has come through the CRISPR technology. Now, what should we be doing now? And one of the reasons why we are having this webinar, number one, is that we should begin to create awareness on CRISPR technology. Like I told you, that one of major setback we have is that so many of our stakeholders, patients, they are not aware of this new technology. So yes, people are hearing about CRISPR, but what is it all about? So you are interested in this CRISPR, you need to venture into it and start creating awareness. In fact, I read in the news, was it yesterday or the day, that in Kenya, in fact, they are even taking the message now to going from religious organizations, because when people see new thing like this, these Oyibo people, they've come again, they want to kill us, because this is science. Science has only one language. Science is universal. If you can discover it here and use it here, so be it. So it is not a white man bringing something. It is what you can discover. It's what you can check yourself. Science is verifiable. You can go to the lab and check it, whether what they are saying is correct. So we need to start creating more awareness on CRISPR. We need to start conducting more research on CRISPR. We need to start seeking more funding for CRISPR, because when we create the awareness, stakeholders, businessmen, the town, they will know that they can bring their funds for us to because it's actually, the research is actually capital intensive. So we need a whole lot, but it's worth it. Then we need a whole lot of collaborations. We have some people that have joined us from other African countries, from Kenya, from Zimbabwe, and all other places. We have people that have joined us from other universities here in Nigeria. We can collaborate, we can work together so that we can share knowledge, share resources, and that can actually help us. And then we can also be an ambassador of CRISPR. That means anywhere you are, you stand as an ambassador of CRISPR to explain to them what it's all about, what are the advantages that they can be. And number 6, we should believe in ourselves in Africa, because in Africa, we need to believe in ourselves that we can do it. It's not a white man technology. It's just the latest technology that we need to improve on it. So we need to work fast on this so that we'll not be lagging behind. Now, what is the major concern? One of the major, I know some people are interested in CRISPR, but one of their major concerns is that they will say, I don't have the training, I don't have the hands-on training, I don't have the practicals, I don't know how to do it. And so they are worried, they are thinking, what am I going to do? Now, we have a solution. Last year, this year, University of Lagos, we organized a hands-on training on CRISPR technology in our lab here in University of Lagos. The second edition will be coming up very soon, where we are going to engage in hands-on and training. And based on our proposal, we are looking at a situation whereby we are going to, last time we used Cas9, we are looking at the possibility of using Cas12 now in our oncoming workshop, hands-on training that you can do yourself. And we can help you design your project if you want a project along any line on CRISPR. That's what we can help you do here in University of Lagos. We can tell you that, okay, this is how to do it. This is how you can use the CRISPR. You can design your project along the line. Okay, in that same workshop, we're also going to demonstrate how you can use CRISPR to edit plants to get desirable quality or quantity, like Professor Fortune told us earlier in his presentation. Do you want it a bit bigger, faster, to grow faster? There are a lot of things that you can play around with CRISPR. And let me tell you that they are safe. You see, the way CRISPR is being done, it's not the way it's being projected. People just thought that you will just put chemical in one thing and then you start selling or you start planting. That's not the way it's done. After editing, there are lots of downstream analysis that you do to confirm that your editing is correct and to ensure that the sequence there is okay. And then there are a lot of precautionary measures that is being taken before some of these things are disseminated into the market. So it's not that we don't just edit something and say, oh, this is another GMO that is coming and everybody's running up and down. So it doesn't happen that way. There are a lot of precautions that happens. So we are going to demonstrate in our workshop how you can also edit some of these plants to get the kind of result that you want. And we're also going to try in that workshop to see how you can also use Cas9 for treatment of a particular disorder. So these are the 3 areas we are going to try as much as possible to cover, all things being equal, to cover in the forthcoming workshop that will be coming sometimes next year here in our laboratory, the Genome Editing Laboratory in Department of Cell Biology and Genetics, University of Lagos. So those are the things we are going to demonstrate to us, arouse our interest, our appetite for CRISPR technology, and see how we can use them to solve Africa's problem. And doing that, what you need to do is that you need to Africanize. You need to Africanize that CRISPR technology. We need to own it. You don't need to see it as a white man work or a white man technology. like I told you, science is universal. They are doing it in Asia, they are doing it in Europe, they are doing it in America. You understand? So they are doing it in Australia. Why should Africa be left behind? Because as it stands now, we are still far behind. The training that was given to us on CRISPR, Dr. Fortune will remember, was being sponsored, you understand, from outside Africa, So when we begin to create some of this awareness and believe in ourselves that we can do it, when we have a lot of sponsor, when we have a lot of buying, we can begin to do some of these things across Africa, and then we can use it to actually solve our own problem. Don't forget that we have a lot of issue of food insecurity in Africa. We have a lot of health burden in Africa. People die every day on diseases that ordinarily should not take life of people. Go and check the life expectancy. The life of expectancy in Africa has dropped drastically compared to elsewhere that are well developed, embrace some of this latest technology. Yes, with a lot of regulations, but then it is worth it that we venture into this particular research space and begin to bring out something. So I want you to look forward to this particular hands-on training that will be coming up very soon. We'll publicize it and make it known to us as the time goes by. This is some of our genome editing team members. You can see their faces after one of our sessions in the lab. We also have some of my students also sometimes they go to after some sessions in the lab, University of Lagos here, after editing some genes in our own laboratory. This is another set of students as well, after a laboratory session also, after we also try to do some knockouts of gene in University of Lagos. So in conclusion, I want to crave indulgence of everybody, appeal to everybody listening to me. This webinar is to raise more awareness amongst us, to call our attention to join in this latest technology, and to let you know that more attention is needed, more funding is needed to be channeled towards us adopting the CRISPR technology in Africa. And this will not in small measure is going to greatly improve our healthcare delivery in Africa so that we can live a fulfilled life. Thank you very much for listening to this topic that I've taken. Thank you very much.
Thank you very much, Professor J.B. Binari, for that exclusive session. It's really a great thing to have, people like this in the academia pulling through to ensure that the latest science is what is being delivered as the case may be. Okay, so for the purpose of those who just joined us, I got information that some of us were unable to join and had to be resent the link. Okay, so but I want to assure you that you've not lost out totally. Okay, so permit me to recap, some of the presentations today, starting with that of our erudite scholar from Zimbabwe, in the person of Dr. Fortune. Okay, so Dr. Fortune took us briefly through the milestones, that CRISPR has undergone from 1987 down to 2014, even till date. And of course, he took us through the status of genetic improvement. And to be specific, he mentioned the milestones that have been achieved, even in terms of having the right development in cattle breeding, in terms of the body weight, milk yield, and of course, in terms of the peak, if we're talking about the peak, he also talked about the efficiency of food conversion in pig. And of course, good egg weight production in poultry. And he didn't even stop at that. He also talked about the improvement that have been done in mice to increase the litter size, also increase the tail length, and of course, strategies for achieving success. Well captioned. So please do not think you have lost out, okay? And to go into the presentation from Professor Minari, okay, which he started with the challenges we have in healthcare. You agree with me that the challenges in the healthcare in Nigeria is nothing to write home about, okay? But we have people working behind the clock to turn around this, and bringing in CRISPR technology is something that would really turn around the healthcare and delivery system. Of course, he talked about the concept of CRISPR system. All of us have had CRISPR. I don't know now, with this exciting lecture from Professor Minari, I want to believe that you've been able to get a little glimpse, if not everything, okay? But I want to be sure, okay? Did we get anything? I want to see your response in the chat box. Yes, I want to see yes, okay? I want to see yes in the chat box. If you know yes, you're able to take in something, okay, from this lecture from Professor Minari. Okay, so I can see a lot of comments now. Okay, some of us are asking for slides. Some of us, okay, I can see the yes coming in. Great, you're making my day. All right, so these are tips of the eyeball. We're going to be doing a lot is coming up, okay? So the workshop is also coming up. Okay, so Professor Minari, well, I can see one of the lecturers from Ilorin is asking for the slide. So Prof, maybe you'll respond to that later. Okay, so he also talked about the advantage of CRISPR technology, which is in terms of fast diagnosis, easy diagnosis, targeted therapy, and of course, accurate diagnosis, okay? He went further to talk about examples of, the application of CRISPR technology, which has been found useful in the treatment of sickle cell disease, going beyond management, but this time around, more targeted therapy, okay? So he didn't stop there. He also mentioned and continued, if I could ask you, in this particular presentation, what did Professor Minari said 1,001 times? He kept saying Africanizing. I want to believe this afternoon we have ambassadors in the house who is ready to Africanize CRISPR, okay? And not to stop there, Professor Minari now manufactured an English for us this afternoon. And he said we need to CRISPRize healthcare in Africa. And I believe we have ambassadors that would help in this move. Okay. In conclusion, he said, what should we be doing in Africa? He said, number one, awareness of CRISPR, conduct research on CRISPR, and he gave his word he could also be of help in this regard. Seek funding, and of course, the third fund and every other funding that is needed. Encourage collaboration, okay? So we can also see, even in this webinar, we have collaborations from Zimbabwe in the person of Dr. Fortune, okay? So be an ambassador of CRISPR, and let's do what? Let's believe in ourselves in Africa. If we believe, yes, the webinar is being recorded, okay? So I can somebody asking if it's recorded. Yes, it's being recorded, okay? So let's believe in ourselves. That's the last note, the last word from Professor Minari. Let's believe in ourselves. And as many as those who are going to believe in ourselves this afternoon, we are setting a pace to see how CRISPR technology will revolutionize health delivery and food production in Africa. So having said that, it is time for question and answer. I have seen one question in the chat box. Do we have any other question? If we don't have, okay, maybe we'll treat the one we have. And that question was directed to Dr. Fortune. Okay, so I'll read it out. So quickly, I'll read that. Okay. Dr. Fortune, are you live with us?
Yes, I'm still with you. Thank you.
Okay, thank you very much. All right, so I would want you to respond to this question from Judith Okanazu. She said, thank you for the presentation, Dr. Fortune. My question, we have seen the power of genetics and importance in livestock production. How do we weigh the risks in comparison to the importance? The mic is yours, sir.
Thank you very much. I think what we usually use and the current framework that is being used for genome editing related research and also commonly in biomedical research, there are regulations that are in place, and most of these activities must or should go through ethics review committee. Names can be slightly different from country to country, but generally there are ethics review committees that are set up and that will have various professionals and lay people to look at proposals so that the research is looked at and either granted ethical approval or not. And what that entails is there's a very close look at what is really the problem that you're looking at, and what is the possible solution that is coming? What is the cost? Is the methodology going to affect people or animals, and to what extent? So there's an evaluation that is done in terms of looking at this research so that there's at least a cost-benefit analysis. And if the risks and damage outweighs the benefits, usually clearances are given. Then there are also general regulatory bodies, particularly for things involving genome editing, whereby even the products themselves that come out, they are not put in the market directly, but there are also evaluation periods that are there, so that before things reach consumers, for example, if it's food related, there is some time for testing and evaluation of those products. So in a nutshell, there is a regulatory and ethical processes involved, and we hope these are adequate, or at least give some security to users and consumers of these technologies.
Great, great. Thank you very much, Dr. Fortune. I believe the person who asked the question, Judith Akanazo, I believe you're okay. Yes, yeah, thank you. All right, thanks for that response, Judith. Okay, so we have a myriad of questions here. Okay, so we're going to take some, and our presenters would go ahead to answer directly in the chat box after now because of our time. Okay, so I will take the next question from Oga Godwin of Omobi. So thank you, sir, for your presentation. It was quite enlightening and interesting. What is the cost of implementing CRISPR technology in a hospital setting? What strategies can be employed to make CRISPR diagnostics affordable and accessible in resource-limited hospital settings? Are there plans on ground to produce CRISPR reagents locally? I believe this question is for Professor Mina.
Yes, thank you very much. Yeah, of course, the CRISPR technology is still very much expensive as we speak. But you see, part of what can actually assist us in making it affordable is actually when we have quite a number of people working in that space. And then that will also help us. And that will also link to the other question you raised. Now, producing some of these things locally, that also depends on us. You understand? So we must first of all embrace the technology so that it becomes common and it become affordable. And how are we going to do that? We'll now begin to do some of those reagents, begin to research. It's also research. So we need so many people that's going to work in different spaces. You know, for example, in the typical molecular biology, that's okay, primer, we order primer, but I mean, I'm happy now that yes, 1 or 2 places in Nigeria now, people are researching on how to produce primers locally, even if it's not as effective as the ones we import yet, but they can improve on it. But at least work is on the way, and that's why we need quite a number of people in the space. So when we begin to produce some of our reagents locally, when we embrace it first, produce our reagents locally, is going to reduce the cost. So it now make it more affordable because we are now able to produce it locally. And then that will now make it also accessible because we have quite a number of us just working in that space. So I'm happy that people are coming from different angles. That can even be the area you will focus on in your own research. Thank you.
Thank you very much, Prof, for that explicit answer. So I see a question here from Oguero Nelson Chibike. But I want to believe the question has already been treated by Dr. Fortune. You said, what are the steps that are being taken to address the issues of ethical concerns arising from gene editing and establishing the robust culturally sensitive regulatory framework? So while he was responding to the first question, he touched on that. But if you are not satisfied with that, please kindly highlight in the chat box. So probably he might need to do that more explicitly. Okay, so I believe this next question is for our first presenter, Dr. Fortune. He said from Oladayo Abidemi. So thanks for the wonderful presentation, sir. The first presenter talked about published work on the use of CRISPR in solving food security problems. Can he please help in sending the link to access the papers and their title? Okay, so Dr. Fortune, I don't know if you are able to do that. If it's all you can do later.
Okay, I'll give links to you, the moderator, then you can share later on.
Yes. No, perfect. Thank you very much, Dr. Fortune. Okay, so maybe this might be, I might take 2 more questions so that we can move to the next session. However, I want to assure you that every question in the chat box and the ones that you will drop after now would be treated by the presenters individually, and you will get the responses in the chat box. Okay. All right. So this is from Temiloluwa Owa. Does tweaking the development / the life cycle of crops, livestock we eat not affect us? Dr. Fortune. So this particular person has directed the questions to both presenters. So he said the first question goes to Dr. Fortune. Does tweaking the development or life cycle of crops or livestock we eat not affect us? And the second one, like autism, which one part of the brain is overdeveloped and another is significantly underdeveloped? What does enhancing the growth hormone in livestock make them redundant in? Okay, so Dr. Ford, I don't know if you got the question or maybe I retake it.
Okay, I got the first part. They were asking about the issue of whether genetically modified food, crops, and livestock will affect us. With the information or knowledge that I have, I haven't seen records or evidence of GMOs affecting people, because most of these things that are being synthesized, either it's meat, then it's cooked, it's brought through the digestive system and digested. Similarly, even plant material. So yeah, that is my response to that one. The second one, I didn't get it quite well.
Okay, thank you very much. So the second one, so this person is asking, okay, like autism, which has to do with one part of the brain being overdeveloped and the other one underdeveloped. So she's asking, okay, what does enhancing the growth hormone in livestock make them redundant in? So I really don't get it very well. But the first one is saying, she gave an example like autism disease, okay, which has to do with the brain not being developed properly, and now went into livestock. So the question is kind of disjointed. Probably Temiloluwa, please, probably you rephrase your question, the second question, so that we can have clarity and you'll have the response. Okay, so Professor Minari, the second question goes to you. Say, if you were to hypothesize the complications resulting from adults having fetal hemoglobin, what would it be?
If I'm to think it.
Okay, if you were to hypothesize, I guess trying to say if you were to create an hypothesis, okay, on the complication resulting from adults having fetal hemoglobin, would it be? What would it be?
Okay. I think the person is suggesting that is there a possibility that there will be a complication with HbF? Well, so far, we've not seen any complications from HbF. HbF functions normally because the function of the hemoglobin is to be able to transport some molecule, which it did initially before it was suppressed. And so far, it does it well. In fact, some of the herbal alternative medicine that people use, they actually increase production of HbF, just help to boost it. So you can correct it that way. And also, by the way, CASGV decided to do his own along that line. We can still decide to focus on correcting the sequence itself that was faulty, and then put the normal base therein, so that you have a complete set of nucleotide that is going to produce the glutamic acid itself. So that one is also possible, and CRISPR is also used on that. Okay, so far we've not seen any record. Just like the question that Dr. Fortune answered the other time on the GMO. You see, we sometimes are just afraid based on, we're just imagining things. But in science, we do more of the hands-on to scientifically prove that whether something is going to have toxic effect or not. And so far, there's not been any scientific proof to show that even some of these GMOs that people are afraid of have any side effect that normal plant is not actually having. And let me tell you that some of these genetic interplays happens normally in the system. The difference between this and that is that this is a controlled one, that is uncontrolled ones. And so with that, you are able to monitor and focus it through to ensure that this is what is going to do. And you also able to monitor it. I told you, over time, it's being checked, okay? And on the issue of ethical approval, that is part of why we are even discussing this also. Our fear is understandably, but it is we, the scientists, are actually supposed to come out to develop the framework of regulation, to develop the framework of ethical issues, okay, so that it will not go above board. You remember in China, a scientist actually edited babies. They believe that it is without their consent. They edited them against HIV, and the babies are still doing fine, even as we speak. So, but we need ethical, I agree that we need ethical regulation, you understand, to make sure that everything works within the ambit and the framework that is going to be beneficial, okay? And that we, the scientists, need to do scientific appraisal to be sure that anything we are releasing, they are safe for us. They are not having long-term effect as we monitor them, as we follow them through, to ensure that it makes us better and not give us worse off. Thank you.
Yeah, thank you very much, Paul. So before you go, the last question for this session, okay? Please, okay, this is from Kaze. Okay, Kaze Ngedia asking, please, Prof, is there a hope to apply CRISPR-Cas12 in the routine molecular diagnostics protocol in cancer management in Africa? As I listen to the presentation, we can use it for early diagnosis of cancers.
Yes. So of course we can use it, but the challenge we have now is that we have few hands working on it. That's why I said we need more of us here to be ambassadors of CRISPR-Cas, so that we can work on different aspects of it. But I can tell you that yes, there is hope, but we are still very, very few. The knowledge is still a little bit low, and even scientists, people just have idea of what CRISPR is all about, but the real practical hands-on on it is still very, very low, and that is what we need to work on to ensure that we have the practical hands-on on CRISPR, and then we are able to deploy it and then bring it out. So there is a lot of hope. We can use the Cas12 for early diagnosis for cancer to ensure that you are able to easily pick it up as early as possible, because of course early diagnosis is going to help in managing, and if possible we have a treatment in treating it completely, and the person can live a normal life. So the hope is there, but it still largely depends on all of us.
Great. So we've heard it. We've heard it from the horse's mouth. The hope is there, and it lies on every one of us. It relies on every scholars in the house, every researchers here present. The hope is in us. A lot of questions, like I said earlier, presenters would go to the chat box and answer every question. Okay, so we have a lot of questions with the next workshop. I would like to be the ambassador and all of that, and that will lead us to the next presentation. Okay, so I will be inviting my colleague who will be talking about the genome editing research group. I know when Professor Minari was speaking, he talked about the genome editing research group. Okay, so it would be nice for us to, okay, so please, can we go to the presentation and welcome somebody who will be taking us on that. Please kindly go on full screen. Okay, so the next presenter will be taking us on GERG. And what is GERG? Genome Editing Research Group in the University of Lagos. Okay, in the Department of Cell Biology and Genetics. And that will be taken by Samuel Idowu Dada. Samuel, the mic is yours. Please kindly go ahead.
Thank you very much. Please, can you hear me?
Yes, we can hear you.
All right, thank you. Thank you so much, everyone, for being here. So briefly, I'm just going to talk about the Genome Editing Research Group here in University of Lagos. So standing on the existing protocol, it's such a delight to welcome you again to this webinar, proudly organized by the Genome Editing Research Group, the Department of Cell Biology and Genetics, University of Lagos. So it's a great time already, and we're delighted as we've explored the topic Africanizing CRISPR technology for food security and better healthcare delivery. As we've all heard, the event is not just a platform for learning, but also an opportunity to connect, to collaborate with each other and ignite new ideas in the ever evolving world of genome editing, as we've done now. So briefly, let me just talk about the driving force behind today's event, which is the Genome Editing Research Group. And this group is led by the visionary Professor J.B. Minari, our second speaker. And the group is at the forefront of the genome editing research in Nigeria. Yes, you heard right. We are at the forefront of genome editing research in Nigeria. We are making significant stride in molecular biology, biotechnology, and genetic engineering. And under the leadership of Professor J.B. Minari, the group has been able to conduct research on microbial genome editing for sustainability, which we've explored how CRISPR-Cas9 can be used to engineer, some bacterial strains for enhanced enzyme production. For instance, by editing a particular gene, we've been able to improve several enzyme productions such as lipase, amylase, cellulase, and many more using agricultural byproducts. And this research demonstrates the potential of turning waste into valuable resources. Also, in the second part of our genome editing research group in the biomedical section, we are working on cancer and some infectious disease using the CRISPR-Cas9 as a functional genomics tools and as a quest for novel, efficient, targeted therapy for this disease, as we've heard from Professor J.B. Minari. And as well, in this amazing group, we've been able to carry out an hands-on training and capacity building. We are committed to empowering scientists and students with the skills to, excel in genome editing. from hosting the first ever CRISPR-Cas9 genome editing hands-on workshop to mentoring researchers across various academic disciplines, areas of research and different levels, from BSc to MSc to PhD. We take pride in building communities of innovators, and our work is as well being driven by deep belief in the power of science to transform life, and we're excited to share some of that passion with you today, which I believe we've learned a whole lot. So about this webinar, the webinar is a testament to what collaboration and curiosity can achieve. The theme, Africanizing CRISPR technology for food security and better healthcare delivery, it reflects our commitment to advancing knowledge and as well, betting out meaningful discussion, as we've heard from our two amazing speakers, Dr. Fortune and Professor J.V. Minari, as they've, dived into cutting edge topic. And lastly, the Genome Editing Research Group is proud to host this webinar as part of our mission, to advance science and empower researchers. And we hope the two sessions have inspired and as well equipped you with all the newest ideas to contribute to the global scientific community. So it's a thank you from our group, and together we can push the boundaries of what is achievable in genome editing and biotechnology. Thank you very much, everyone.
It's less than
Thank you.
Please let's mute our mic. Please kindly mute your mic. Thank you very much, Samuel, for that brief introduction to Genome Editing Research Group. Okay, you want to hear more, you want to know more, please kindly reach out to Professor Minari. Okay, so at this point, it is time to say goodbye. And we have in our midst one of our researchers and also a member of the Genome Research Group too, here in the University of Lagos, also a lecturer in the Department of Cell Biology and Genetics, in the person of Dr. Abazu Uche, who will be taking the vote of thanks to our distinguished participants this afternoon. Dr. Abazu, the mic is yours.
Okay, thank you very much, Dr. Ogo. Thank you. That was a very beautiful encourage. And I want to say thank you to every one of us that joined from across the globe, wherever you have joined us from. And I believe that each and every person has gained a lot from this webinar. Thank you for your time. Thank you for your resources. We will not go without saying a big thank you to the chief host, the Vice Chancellor of the University of Lagos, Professor Ugushola, and of course the co-host, our own amiable Dean of the Faculty of Science, Professor Shekun, and of course the host, the department, the HOD of the Department of Cell Biology and Genetics, Professor Ogunkomi. Thank you so much for having us. Thank you for all the effort that you put in to make this webinar a success. We will not also go if we don't say a big thank you to our speakers. Our speakers, I will start by saying thank you to Dr. Fortune. Dr. Fortune, that was a beautiful presentation. They will say thank you so much for your presentation. Thank you to Professor Minari. Thank you for that beautiful presentation as well. We also want to say a big thank you to all the members of the organizers. Without your effort, without your little contribution, this webinar wouldn't have been a success. For all those that work at the background, thank you so much. We recognize also all our sponsors. Thank you so much. We also want to appeal and continue to say thank you. Please, if you have anyone who can sponsor us, just like Professor Minari has rightly pointed out, sponsorship. We really need this. As we are saying thank you, also asking for sponsorship. And on the last note, please, Africans, we must believe in ourselves. Africans, we must work hard. Africans, we must be about a good ambassadors. Because if we don't believe ourselves, then we are going nowhere. But the first step is believing in ourselves, putting the effort together, and we can conquer. Thank you for being on board. God bless us as you have a lovely day and a lovely weekend.
Thank you.
Okay. Thank you very much, Dr. Uche. We also appreciate you for being part of us and for your assistance. Thank you so much. So please, announcements. I know quite a number of people have been asking concerning the hands-on. We are going to get back to us and send the flyer, the notice when the hands-on training is going to come up. And then we'll let you know when it's going to come up and the modalities that is going to be involved in it. So once again, we'll appreciate everyone, and we look forward to also you attending our subsequent webinar. We'll still try to organize. So the importance of it is just to keep the discussion alive, keep the interest up there, and ensure that we are able to take it around, especially with the scientists. Thank you very much. I also recognize the presence of Dr. Shifao. Shifao is a geneticist, has also been part of us since when we started, and all other our staff members that also joined us that we have not mentioned. Thank you. Have a blessed day till we see again. God bless you.